Okay, we'll go ahead and continue with the lecture series on microbiology, and we'll start with the microbial mechanisms of pathogenicity. And remember from the previous chapter, pathogenicity is referring to the disease-causing ability of certain microorganisms. So by definition, pathogenicity refers to the ability to cause disease in a host. We're talking specifically about microorganisms now. First, however, the pathogen must enter the host body somehow. Virulence is a term that refers to the degree of pathogenicity. We've already alluded to in earlier lectures about certain things that make organisms more virulent. Some organisms have a endospore, which protects them from environmental insults. Other organisms secrete toxins that can proliferate or aid them in their ability to cause disease. So those are virulence factors. So the avenue by which microbes gain access to the body is referred to as the portal of entry. And that's another term that you need to be familiar with. Just as a review, pathogenicity, ability to cause disease, and virulence, the extent of pathogenicity. So how can the microorganisms enter the host? Well, through the mucous membranes. That's one area. You can gain access to the body. Pathogens can penetrate the mucous membrane, the linings of the conjunctiva of the eye, the linings of the respiratory system, the lining of the gastrointestinal system, and the lining of the genitourinary system as well. Those are common areas where we'll see microorganisms enter the human system. The respiratory tract is the easiest, most frequently used route to enter into the body by infectious microorganisms. Diseases contracted by this route are as follows, such as the common cold, pneumonias, tuberculosis, influenza, measles, and even diseases such as smallpox, though we haven't seen any cases of smallpox in our country since the 1960s, certainly because it's transmitted by droplet transmission. We see that as an infectious organism that can pass through that route as well. Microorganisms can be contracted from food, water, from the fingers, so poor hand washing technique can play a role. Either way, these organisms can enter the body through the gastrointestinal tract. Many of them can be destroyed by stomach acids. In fact, many of the microorganisms that we inhale into the respiratory tract get trapped in the mucociliary protective layer of the lungs, coughed out, and either expectorated in sputum, or either we swallow it. And as we swallow it, it goes down the gastrointestinal tract. The powerful digestive enzymes in the intestinal tract will kill those organisms. However, there are certain diseases such as poliomyelitis, hepatitis, forms of hepatitis, hepatitis A specifically, typhoid fever, amoebic dysentery, shigellosis, bacillary dysentery, and cholera that are transmitted via this manner. Examples of pathogens that enter through the mucous membrane include the genitourinary tract, such as HIV disease and AIDS, chlamydia, syphilis, gonorrhea, and so you can see all of the sexually transmitted diseases can certainly enter through the through the genitourinary tract. However, some of these same organisms can also be transmitted orally as well. In aspects of oral transmission, we see oral sex as one of the ways that these organisms can be transmitted, and so you can get gonorrhea, syphilis, and or chlamydial infections in other locations of the body around the head and neck. The eyes can be infected, the oral mucosa can be infected as well. But again, this is a mucous lining. Skin with few exceptions, such as hookworms, microorganisms, cannot penetrate the skin unless the skin is broken. Some fungi can exist on the skin and can cause tinea infections, tinea capitis, tinea corporis. These are infections related directly to fungal elements. We've already talked about candidiasis in earlier lectures. We can see candidiasis can occur on the surface of the skin. Common name, manilial infections related to fungus. But fungi can grow on the keratin of the skin. Other microorganisms can gain access by penetrating openings in small hair follicles or down into the sweat ducts and glands. Parental route. Parental refers to injection. So we see the skin or the mucous membrane can be punctured or injured or traumatized. Microorganisms can gain access to body tissue through this route as well. The route is referred to as the parental route. And so common portals of entry, and that's the term, portals of entry, mucous membrane, skin, or a parental route. The common organism for parental route, too, I may have said this before, sporotrichosis. It's a fungal infection gardeners get that working in the garden, they get pricked by a thorn bush, a rose bush, and get injected with a fungal element that can cause infection. Preferred portals of entry. Whether or not the disease results from the entry of a microorganism depends on several factors. Organisms must enter by preferred routes. So, for example, Salmonella typhi has to enter through the gastrointestinal tract, and it will cause typhoid fever, but it can't be rubbed on the skin and transmit infection. Clostridium tetani has to penetrate the skin, causes tetanus, and Corynebacterium diphtheria enters through the respiratory tract and will cause diphtheria. Measure of virulence is referred to as the LD50. That's the lethal dose 50, which is the dose of the pathogen that can kill half of the tested animals. Take 100 lab rats, and if you want to find out what the virulence factor is for an organism, you infect the organism, and whatever level of organism, bacterial load or viral load, you have to give those rats, that 50 of them will die. It's called the LD50. It's a simple way to relate it. So if pathogen causes only a non-fatal disease, the lethal dose is referred to as ID50. So infectious disease, 50 of them got infected. That's referred to as the ID50. LD50 is if 50 of them died. The fewer organisms required, the higher the organism. So if you only had to have a small, only 10 colonies of E. coli were infected lab mice, and 50 of them died, so 50 colonies is your lethal dose. But if you want to get a virulent form of E. coli, say E. coli 015787, then you gave only 2 colonies of that and 50 mice died. Then you know the virulence of E. coli 0157 is much stronger than what you see E. coli. I'm just using that as an analogy now. It's not that E. coli would infect the rats and kill them at that level. But it gives you some reference point of what we mean by LD50 and how that relates to virulence factors. ID50 and LD50, again, infectious dose, 50% of the tested population affected. LD50, lethal dose, 50% of the population tested. For most pathogens, adherence necessary for pathogenicity. So the attachment between the pathogen and the host makes use of surface adhesions and or ligands and complementary surface receptors on the host cell. Adhesions also mostly glycoproteins or lipoproteins, which are frequently associated with structures such as the fimbriae that I showed you on some other organisms in earlier lectures. Now, recognize that some other factors that can lead to attachment, we talked about this with viruses, and you've studied this for the test coming up on Wednesday, is the spikes that viruses have. Viruses can use the spikes for purposes of attachment as well. Many microbes have the ability to form a biofilm. These are masses on the surface that secrete a glycocalyx and aid in attachment. Within the biofilms, they share available nutrients, relatively protected from antimicrobials and the immune system. Now, biofilms, just as an aside, this is not testable material, but biofilms can develop and not lead to disease as well. For example, water pipes. Water pipes typically have a biofilm inside the pipes. This is the normal flora of a water pipe. This is a film, or residue, that kind of develops inside the water system. Now, it doesn't release a bacterial load into the water system that causes disease, but the biofilm is a normal part of the ecology of a water system. Now, what happens in conditions when the water becomes stagnant? This is why water has to keep flowing, because it keeps the ecology of the pipe the same. But if water becomes stagnant and allows this bacteria to grow out of control without continually being flushed through the system, then what was a diluted biofilm then becomes a concentrated biofilm and can lead to disease and some problems. And you see this constantly in the recovery phase in disasters because the water system or the sewage system gets compromised due to the effects of whatever the disaster was. So the water system doesn't flow, or they have to turn off the water system, or the sewage system, even for that fact. The sewage pipes develop an ecology in the pipe as well. And then you get a certain number of bacteria will begin to grow out of control, and what was a non-pathogenic type of organism now turns into a pathogen because of stagnant water. So water has to keep flowing to keep the ecologies of these piping systems the same. Other organisms that can infect human system, Bacillus anthracus, also known as anthrax, can penetrate the skin, can penetrate the lungs through inhalation, can also penetrate the gastrointestinal tract through ingestion. So you can see three presentations of anthrax: skin, inhalation, and ingestion. There'll be an extra credit opportunity coming up for you before the end of the semester where you'll be able to see the effects of these types of diseases and some of the adverse consequences and signs and symptoms of infectious organisms, Bacillus anthracus. Adhesions, ligands bind to receptors of host cells. You see again the glycocalyx, the fimbriae, M proteins, OPA proteins, OPA, not Oprah, OPA proteins, and the tapered end of organisms that can help in attaching and leading to infection. So the tapered ends you see in Treponema pallidum, the causes of gonorrhea, Neisseria gonorrhea, the OPA protein, and then M protein, strep, fimbriae, E. coli, and then the glycocalyx, important, and strep mutans, which we'll see when we talk about dental diseases or oral diseases later on in the lecture series. Capsules. For many pathogens such as strep pneumonia and Haemophilus influenza, capsules confer resistance to phagocytosis. Cell surface sometimes contributes to the invasiveness. Strep pyogenes, for an example, contains a protein referred to as an M protein on the fuzzy layer of the fibrils, the cell surface that help resist phagocytosis, which will improve adherence. So if you can adhere and then also can fight off the process of phagocytosis, then obviously these organisms would proliferate in the human system. Waxes in the cell wall, such as in Mycobacterium tuberculosis, can resist digestion by macrophages. The reason why tuberculosis can be such a bad disease in humans is once this organism gets in the lungs and the macrophages can't get rid of it, what happens is the body, by its immune response, will start to wall off this organism. So if you can't get rid of it, one of the things that the body will do is just produce a lot of inflammatory cells and surround the infectious element. Now that sounds like a good thing. The only thing is, because it occurs in the lungs, if you start to put all these inflammatory cells and it begins to wall off these organisms, it takes up the room in the lungs. And we need the room in our lungs for air because that's how we breathe. And so if you get an infection with tuberculosis, what happens is you develop these huge lesions. They refer to them as cavitary lesions because our immune system is trying to wall off the infectious organism. It will lead to scar tissue. Real bad cavitary pulmonary tuberculosis, however, the patients have significant respiratory compromise, again, because the organism has a waxy coat and it prevents digestion through this process of phagocytosis. Enzymes, there are extracellular enzymes referred to as exoenzymes that have the ability to break open cells and dissolve material between cells that forms or dissolves blood clot. These can contribute to the invasiveness. So imagine an organism that enters into the body. And again, some of our initial defense mechanisms are the skin, and then even if you break the skin, at least it would only be a local or a focal infection because there are tissue layers that help to localize an infection. But what if the bacteria secretes an enzyme that can destroy tissue? And so it secretes an enzyme, it destroys tissue, and thus the bacteria can migrate in further areas and disseminate through the body because of the coagulases, or the ability of some of these organisms to produce enzymes that will destroy tissue. Coagulases, for example, can cause the destruction of particular types of fibrin clots. And coagulases actually have been used, though clinically, purified coagulase forms, enzymes, been used clinically. Streptokinase was used to be the most popular agent that was used in emergency rooms to clear blood clots in patients with coronary artery disease. Blood clot blocks off the coronary artery, you inject some streptokinase, it goes to the side of the clot and it breaks the clot up. So it can be used in some therapeutic managements, but as we talk about it today, we're only talking about the pathogenicity of these organisms. Bacterial kinases are enzymes that break down fibrin and dissolve clots, forming the body tissue. where the body has attempted to try to isolate the infection. Streptokinase is what I've just alluded to here earlier. Hyaluronidase is an enzyme that's secreted by certain bacterium that digest hyaluronic acid. Compounds is a mucopolysaccharide that holds together certain body cells, such like you would see mortar hold bricks together, especially connective tissue. Both the organisms can produce gas gangrene, which is a number of streptococci can produce this enzyme, which will promote the spread of infection through adjoining tissue. Coagulases are produced several species. Clostridium are produced by several species. Clostridium breaks down the collagen framework in muscle tissue. As a defense against adherence of pathogens in mucosal surfaces, the body produces a class of antibodies known as IgA antibodies, and some of these pathogens have the ability to produce enzymes called IgA proteases that would destroy these antibodies, thus, again, allowing these organisms to be more pathogenic. Now, coagulases, again, are another agent that we see clinically, and again, those of you who go on to work clinically may see the use of certain types of coagulases. Elase is a coagulase that's used in the clinic. Patients have decubiti ulcers, or patients may have eschars. Eschars are really thick scars, necrotic scars that typically have to be surgically debrided. If the person's not a candidate for surgery, you can apply this topical coagulase, which will basically dissolve that scar tissue. Scar tissue is a bad thing when it keeps normal tissue from healing. Many of you may have had a scab before from a cut or an abrasion, but the scab eventually falls off because the normal skin can grow under the scab. In a bad scab, in an eschar, what happens, it forms down in the matrix of the tissue. And under the scab, under the eschar, is necrotic tissue, tissue that will not heal up. And so because of that, the person has a long open wound and it won't go away. And one of the ways to get rid of an eschar for a person that can't have surgical debridement done is to apply a collagenase, which again is a specific type of enzyme that can break down a tissue. And basically you apply this over several weeks. This is a long-term type process, but eventually the collagenase will destroy that hard necrotic tissue because it can destroy tissue. Now imagine putting that on normal tissue. One of the precautions we warn about patients that use collagenase is to not put that on normal tissue because it will also destroy normal tissue. A major factor in the body's defense is specific resistance in the immune system. As I alluded to earlier, since the IgA antibody can be destroyed by certain types of enzymes in the body, the immune system that recognizes antigens by producing antibodies specific against them, as IgA antibodies can do. Some organisms can continually change their antigenic structure. Now remember, antigens are foreign invaders. It's something that's not supposed to be in the body. And so the body typically will form antibodies to a specific antigen. But what happens if the antigen changes on a regular basis? Then the body is producing antibodies, but when the antibodies go to attack the antigenic agent, the foreign bacteria or viruses that's in the body, it's changed. And so now the antibodies aren't specific for that. And so because we see this antigenic variation, some organisms can continually change, and it means they will evade our immune system. Another thing that organisms can do is something called mimicking. Some infectious organisms, especially some viruses, can appear just as our normal tissue does. Now, in many of these cases, the organisms can evade the immune system because it appears to be just as our normal tissue would be. In other cases, such as in post-streptococcal glomerulonephritis, that's a condition where you get strep throat, you get treated for the strep throat, 10 days, your throat feels better, but you start to run a low-grade fever, begin to have pain in your back, may even start to see a little blood in the urine, and then what's happened is the streptococcal infection is now disseminated. It's gotten to the bloodstream, turned into a bacteremia, and spread to the kidneys, and now the streptococcal infection is in the kidneys. But the antigen-antibody complexes that join together will get into the membranes of the kidneys, and many of those organisms can mimic or appear to be just as the normal kidney tissue does, and our immune system begins to attack our own kidneys. So this can cause an autoimmune type of a response, and so many of these organisms have that innate ability to mimic our appearance. Just as a recap, the enzymes coagulases, kinase, hyaluronidase, collagenases, IgA proteases All are types of enzymes that will go to destroy our primary defense systems. Siderophores and antigenic variations alter the surface protein, or acidophores take iron from host iron binding proteins. Remember, our system demands iron to be able to carry oxygen. We have to have iron in our red blood cells to be able to carry oxygen. So if you take iron from the host iron binding proteins, our oxygen carrying capacity drops significantly, compromises the immune system, compromises the whole host system, and leads to further infections. So, microbes sometimes can attach to the host cell by adhesions. This activates factors in the host cells in the cytoskeleton, which result in the entry into the cells by microbes. A major component of the cytoskeleton is proteins called actin. Some pathogens, such as Salmonella, produce surface proteins called invasins. These arrange nearby filaments in the cytoskeleton. This causes the pathogens to move into the cells. Once inside the host cell, some bacteria can use actin to propel them from one host cell to another. Actual movement from one cell to another involves a glycoprotein called cadherin. Siderophores, iron is required for growth of pathogens, and so there's little free iron in the host body. And so because of that, as I alluded to earlier, there are substances that can be produced by pathogens referred to as siderophores. These proteins that remove iron from the iron transport proteins that bind more tightly. So iron can also be released from the cells of the host when the cell is killed by the toxins. So again, this relates to the pathogenicity of certain organisms, but it also will compromise the the host system. The immune system requires oxygen, as do other important networks in the body, and so if you can compromise the iron carrying capacity of the host cells, not only do the bacteria use the iron, or the invading organisms can use the iron, but it also compromises the host system even more. Also, as we've talked about before, are certain toxins that can be produced by these organisms. Toxins are poisonous substances produced by certain microorganisms. The capacity to produce them is called toxigenicity. The presence of toxins in the blood and in the lymph are referred to as toxemia. Exotoxins are proteins secreted by bacterium, mostly gram-positive, into surrounding medium released by and following lysis. They are highly specific in their effect on the body tissue. They're among the most lethal substances known. The body can produce antibodies such as antitoxins that provide a degree of immunity to certain exotoxins. Exotoxins can be inactivated by toxoids and used to stimulate antitoxins in the bodies against diseases such as diphtheria and tetanus. And you all are probably familiar with tetanus toxoid. You certainly received one early in life, and many of you may have already received booster injections of tetanus somewhere along the way from a cut or a scrape. If you haven't, you should, and before you start working in the hospital, you certainly should, because tetanus can lead to pretty significant disease. But you receive a tetanus toxoid is what you receive to counteract toxins once exposed. Exotoxins groups are grouped into three principal types. AB toxins, type 3 toxins, named because they consist of two parts, an A part and a B part. They kept it simple. Most exotoxins are of this type. They after you see the release of the toxin from the pathogen, diphtheria toxin is a good example of that. Part B will bind to the host cell, and part AB toxin is then transported into the cell, and their components will then separate as A part inhibits protein synthesis, which again, you know, our cells, that's what the nucleus does. It produces a sequence of events that leads to protein synthesis. If we don't have protein synthesis going on in our cells, then we die. And so you'll see that this will kill the cell. We also can see certain organisms with toxins that will disrupt the membrane. Membrane disrupting toxins, or type 2 toxins, cause lysis of the host cells by disrupting the plasma membrane. Examples are cell lysing exotoxins of Staphylococcus aureus. These toxins will contribute to virulence of killing the host cell, especially the phagocytes and the macrophages, which is that first cell line, the first wave of cells that we see enter into the system and destroy the body's defenses. Membrane disrupting toxins are called leukocidins, white blood cell killers. White blood cells are an important part of the immune system, as we'll see when we talk about the immune system. Streptococcus produces hemonolysins. These are called streptolysins, such as streptolysin O, named because it oxygen, in the presence of oxygen, you'll see it diminished. Streptococcal hemonolysins is stable in the presence of oxygen. It's called streptolysin S. So two different types, one destroyed by oxygen, one that can continue to work its effect even in the presence of oxygen. These hemolysins kill both red blood cells and white blood cells and many other cell lines as well. So you can see where these toxins are really serious. And again, a lot of streptococcal infections produce these hemolysins. Their superantigens are type 1 toxins. These are antigens that provoke a very intense immune response. Stimulated by the proliferation of the immune system cells, it's called T cells, important factors in the body's specific immune defenses, and it's discussed fully in the following chapters. The T cells, in turn, will release an enormous amount of cytokines, and we'll talk about those more when we get into chapter 17. Cytokines, which are proteins that stimulate or inhibit many cell functions. They're released by superantigens. Stimulation can cause many symptoms of disease and even death. Staphylococcus, or staphylococcal toxic shock syndrome, was an example of that. We don't see toxic shock syndrome as often anymore, but certainly notable exotoxins produced there. Exotoxins are also named for the basis of the tissue that they affect, such as neurotoxins, nerve cells, cardiotoxins, heart cells, enterotoxins, the lining of the gastrointestinal tract, and cytotoxins, a wide variety of body cells. Diphtherotoxins, AB toxin produced, we already talked about AB toxins, produced by the Corneobacterium diphtheriae when it's infected by lysogenic phages. We talked about phages, little elements that can live inside of bacteria. They carry a tox gene and will lead to a lysogenic conversion, as you studied in chapter 13. Erythrogenic toxins are produced by Streptococcus pyogenes and produces the red skin or rash characteristic of scarlet fever. Botulism toxin is produced by Clostridium botulitum. This is a neurotoxin that can lead to paralysis, flaccid paralysis when ingested. If you remember from A&P Part 1 how botulitum toxin works, if you remember acetylcholine attaches to receptors in the motor end plate and helps to conduct the action potential, that in the presence of calcium. So you have to have acetylcholine, which is a neurotransmitter. Botulitum toxin will bind to the receptor site. If you bind to the receptor site, then the toxin holds on to the receptor site and you can't get acetylcholine. If you don't get acetylcholine at the point of the cell, then you get paralysis because you have to have acetylcholine. So botulitum toxin can lead to major muscle paralysis and eventually death because the muscles of respiration, such as the diaphragm, is affected by this neurotoxin. Then the person will not survive. Tetanus toxin produced by Clostridium tetani, also known as tetanospasms because of the severe spasms that it can cause. AB neurotoxin that blocks the relaxation pathway of muscle control produces spasmodic contractions. Vibrio enterotoxin, cholera toxin produced by Vibrio cholera. It is a AB toxin that causes release of large amounts of fluids and electrolytes. The results are a severe form of diarrhea. Heat labile toxins produced by strains of E. coli have also identical action. So you'll see very significant diarrhea produced from these types of organisms. Staphylococcal enterotoxins produced by Staphylococcus aureus can affect the intestines the same way that you see cholera toxins. And when you do lab 6, and I changed that for the exam. Sorry, I put lab 6 on this exam. It should be on the next exam, so I'll take those lab questions. I think you get 2 or 3 lab questions there from that. But so if you haven't done virtual lab 6 yet, the whole presentation is about the enterics, the vibrios, the choleras, the dysenteries, Shigella, Salmonella is all discussed there. You'll study about the toxins that are involved in that as well. So just as a review, toxins, substances that contribute to pathogenicity, Toxigenicity, the ability to produce a toxin. Toxemia, the presence of toxins in the host bloodstream. Toxoid, an inactivated toxin used as a vaccine. And then antitoxins, antibodies against specific toxins. Now, remember, toxoids, inactivated toxin, and it's used as a vaccine. So our body, if it's exposed to the covering of the toxin, remember these toxins have a polysaccharide or glycoprotein covering, our immune system still recognizes that covering as foreign to the body. And so our immune system will develop immunities to that foreign invader, even if it's just a toxin. Same thing can apply to antitoxins. These are antibodies against a specific toxin that forms in our body. And so it's an important part of the immune system, and we'll talk about the immune system in just a little bit. Exotoxins, again, just as a review, again, exotoxins produced by cells or released into the system can cause a number of the signs and symptoms that you see. This illustration is out of your book, just basically showing the production of the enterotoxin, the release of the enterotoxin, how the enterotoxin binds to, I'm saying exotoxin binds to the cell surface, their cell surface receptors, and then the component of the exotoxin will alter the cell functioning, typically by inhibiting, and this is the key point, typically by inhibiting protein synthesis. Protein synthesis, once you break the machinery of protein synthesis in our body, then the cells will be inactivated and die. Superantigens, again as a review, type 1 toxins cause intense immune response due to the release of cytokines. Fever, nausea, vomiting, diarrhea, shock, and or death can occur. And then exotoxins, membrane disrupting type 2 toxins, lyse the host cells. They make protein channels in plasma membranes, leukocytins and hemolysins, and this thus will disrupt the cell membrane, breaks the phospholipid bilayer. And remember, when we talked about the phospholipid bilayer, we said the importance of that in human cells and in some microorganisms as well, but here the exotoxin is attacking the human cells. We'll see disrupting the phospholipid bilayer will allow the environment outside the cell to enter inside the cell, or the environment inside the cell to go outside the cell. In essence, it destroys the cell. So the other category is enterotoxins. Now, endotoxins that are not secreted by bacteria but are a part of the outer portion of the cell wall of gram-negative organisms. Now, you will find that gram-negative infections inside the human system are really tough infections to treat. They cause a person to get sick right away. Now, you don't see that many organisms that proliferate and cause the serious illness that these can in a short period of time. Specifically, there are lipopolysaccharides or LPS portions of a lipid A. It's called, recall that exotoxins that we talked about in protein. Endotoxins are released upon death and lysis of the bacteria, as well as during bacterial multiplication. They stimulate macrophages and release damaging cytokines. One of the symptoms of fever caused by interleukin-1, IL-1, and we'll talk about that again more when we get into the immune system, formerly called endogenous pyrogens. Another symptom is shock. Shock's a dangerous loss of blood pressure related to disease. Now, didn't say loss of blood. And some people equate shock. In most instances, you do get shock from a significant loss of fluid or blood in the body. You get in a bad car accident, you lose a lot of blood, you go into shock. But the problem is, well, that is a problem. But on the other hand, when we look at infectious disease, it's not that you lose blood volume, it's you lose blood pressure. Because what happens is these enterotoxins release such a severe and innate effect on the human body that it causes the body's blood vessels to vasodilate. The blood vessels dilate, the pressure goes down. Think about a water pipe. You got so much water in the pipe. If you make the pipe bigger, the water pressure goes down. And so it's not that the blood volume changed, it's just the size of our blood vessels changed. It drops our blood pressure. We have to have pressure to be able to get blood to the brain. We have to have pressure to get blood to the kidneys, to other areas of the body. And so if we decrease the pressure, then we start to underperfuse the rest of the body. And so enterotoxins cause septic shock. And when that occurs, the body vasodilates, inadequate tissue perfusion occurs, and the person will die. The disease is called septic shock and can be very dramatic. It evolves over time in relationship to the infectious disease. But remember, certain infectious microorganisms, such as gram-negative organisms, proliferate very quickly, and because they have this endotoxin, it leads to significant compromise to the host system. Laboratory test for endotoxin is called the LAL test, Limonus amoebocyte lysate assay. This uses amoebocytes, white blood cells, from a horseshoe crab, the Limonus polyphemus, which the enterotoxin can cause to clot. Again, you don't see this in every lab. Now, another term that you need to be familiar with is portal of exit. Portal of exit. And we talked about portals of entry, but now let's talk about portals of exit, and then the reason why portals of exit are important. Portals of exit, like portals of entry, are often characteristic of a disease. Most commonly, these are respiratory or gastrointestinal tract and are important factors in disease transmission. Another example of sexually transmitted disease is the genitourinary tract. Several diseases caused not only by pathogens are lysogenized, but we also see carriers or particular plasmids as well. The cytopathic effect, again studied in chapter 13 or 14, I can't remember now. Cytopathic effect of viruses, visible damage to the host cell. This is referred to as cytopathic effect. You can have a cytal effect. This actually kills the cells, or a non-cytocidal effect, where there is damaging, but it's not lethal to the cell as well. Cytopathic effects for viruses, they can produce one or more of the following cytopathic effects. They can cause a macromolecule or macromolecular synthesis within the host cells to stop, and also cause lysosomes to release their enzymes inside the cell. If that causes, remember, the lysozymes are powerful enzymes that typically are used to digest invading bacteria or viral elements. Can also be used to destroy or break down products that have been used inside the cell. Well, if these lysosomes break open and spill inside the cell, then it will kill the cell. Kill enough cells, you kill the human body. Inclusion bodies, these are granules that are composed of viral parts that can be found in virus-infected cells. Adjacent infected cells fuse to form what's called a syncytium. This is a very large multinucleated cell. Changes its function and otherwise no visible changes. For example, the production of hormones may be produced or may be reduced. We also see cells produce interferon that protects neighboring unaffected cells from infection, antigenic changes that elicit a host immune response, chromosomal changes that induce the possibility of oncogenic viruses or cancer-causing viruses. You see transformation or transformed cells, so it loses its contact inhibition, which leads to an unregulated cell growth. Now again, we talked about that just a little bit in chapter 13 as well. Oncogenic viruses, oncogenic RNA viruses, and some oncogenic DNA viruses. The main difference is reverse transcriptase being in the RNA virus. You see that what happens, especially in oncogenic viruses, is it alters the cells. Typically when cells grow, if they touch each other, they stop growing. But in cancers, there's no contact inhibition. And so these cells will continue to grow, continue to proliferate, continue to multiply, and eventually they stack on top of each other and they form tumors. Or if it's a blood cancer like a leukemia, though leukemias aren't oncogenic viruses, so don't misunderstand what I'm saying, I'm just giving this as an example. In leukemia, certain cell lines will grow out of control. Leukemias, white blood cells grow out of control. And they grow and they grow and they grow and they crowd out all the other normal cells, red blood cells, platelets, all the other cell lines get crowded out by white blood cells in leukemia. And so your body can't function without red blood cells, platelets, or whatever. And so, but all of those types of cells have lost contact inhibition. Otherwise, if the cells touch, they would stop growing. So we can see the antigenic changes that can be elicited in the host response. Chromosomal changes induce the possibility of oncogenicity. or cancer-causing ability. Transformed cells, again, lose contact inhibition. These are some of the viruses we see cytopathic effects: poliovirus, parvovirus, adenovirus, arabidovirus, which can lead to rabies, cytomegalovirus, measles, polymervirus, and lentivirus for HIV disease, which primarily destroys the immune system cells. Some other pathogenic properties in other organisms: fungi, protozoa, helminths, and algae. Some protozoans and helminths grow on host tissue, causing cellular changes to the host. Their waste products contribute to symptoms of disease. Some algae can produce a neurotoxin. Some toxins are associated with fungi, such as ergot. In ergot, we see used now therapeutically in some agents for the control of migraine headaches that reduce muscle spasms or vasospasms. Ergots contain sclerotia, which is a resistant mycelia. We also see ergotism as a result of hallucinations. We also can see it can constrict blood vessel capillaries. It can also lead to gangrene in certain extremities. Aflatoxins are produced by the mole Aspergillus flavus, can cause cancer in the liver of animals. Toxic mushrooms, Amantia phalloides, contain dangerous mycotoxins, phallodoin, which is mantidine. We also can see some algae can produce a neurotoxin referred to as saxitoxin, and we talked about saxitoxins as agents that we see ingested by organisms or fish, sea life, which ingest some of the infected algae, will feed on saxitoxins during red tide, and if you ingest these organisms, these crustaceans, then you can, or fish, you can develop neurological systems accordingly. So, pathogenic properties of fungi, just in review: fungi waste produced by may cause some symptoms. Y'all can see chronic infections that promote an allergic response. There are other toxins that will inhibit protein synthesis: ticosines, fusariums. There are proteases that are produced. There are certain organisms that will produce substances that will prevent phagocytosis, and then certain toxins, such as in ergot, will produce toxins that can lead to vasoconstrictions and other complications inside the human system. Aflatoxins, as produced in Aspergillus, mycotoxins, and the neurotoxins, phalloidin and amantadine, can also be produced by fungi as well. For protozoas is the waste products that can cause a lot of the symptoms. You avoid the host defenses by growing phagocytes. We also see antigenic variations with these organisms. They do utilize the host tissue. The presence of parasites interfere with the host functions. Parasites, metabolic waste, causes a majority of the symptoms. And then we see, again, we talked about with algae, the dinoflagellates, the saxitoxins ingested by shellfish, and then you have a paralytic shellfish, which can lead to poisoning. Also, portals of exit includes the respiratory tract. Again, many of the droplet infections, so sneezing and coughing, can be a portal of exit from a diseased reservoir, human body that can carry disease. Gastrointestinal tract through saliva and feces. Remember, the feces passes and gets into the groundwater, or just by oral fecal route, not good hand washing, and can contaminate and recontaminate an individual or other people. You can also see the urine, vaginal secretions, skin, blood, especially when you talk about biting arthropods. Again, I know you studied that for a test coming up. Biting arthropods like fleas, ticks, mosquitoes. And then remember, houseflies are not biting arthropods, but they do walk in the contaminated areas, contaminated debris, and can spread especially a lot of foodborne diseases because because they land on food that hasn't been eaten. Needles and syringes, fomites that can cause can be portals of exits as well. You give somebody a shot, or what we see in emergency rooms and in clinic settings is individuals that share needles. Again, the blood is being shared because even though the needle is a fomite, the infectious organism still lives in the blood, and so the sharing of needles can also be a big portal of exit as well for certain individuals.